Bismillahirrahmanirrahim. Now this is a mock test which I've made questions from 5090 and 0610, but they cover all the syllabus of both the two syllabuses, 5090 and 0610. So just some difficult MCQs which I have put together so that you all can get a good practice for the revision. And let's continue with the questions now. Starting with question number 17, the graph shows the effect of light intensity on the rate of photosynthesis. So you've got rate of photosynthesis here, which is the dependent variable, and light intensity, which is the independent variable. So say you have one 100-watt bulb, then two, then three, then four, and then five, and so on. And the rate increases by 10, then it increases by 20, then it increases by 30, whatever 30 oxygen produced or whatever. So the rate of photosynthesis can be measured either by saying, okay, volume of oxygen produced per unit time, or the number of bubbles of oxygen produced per unit time, or the rate of carbon dioxide uptake. If you have a closed environment and you have a very fixed amount of carbon dioxide, then that uptake could be, or you can have the mass of glucose produced, but then remember, glucose is converted to starch. So mass of starch produced per unit time, or in 10 minutes, or 20 minutes, or 30 minutes. So now it says which environmental factor is limiting the rate of photosynthesis at X on the graph. You see, at X on the graph, what is happening is that the graph is increasing. So we had light intensity 1, so rate was this, then the light intensity is increasing and the rate is increasing. So if anything, if we are increasing the light intensity, you can see from here, as we have increased the light intensity, the rate has increased. So what is the limiting factor would be light intensity. Now, always remember in such graphs in which there's the rate of photosynthesis, light intensity, whatever is on the x-axis, is the limiting factor from here to here till the graph is increasing. But once the graph has leveled out, then light intensity now is no longer the limiting factor, and some other factor is limiting. And you have three things which limit the rate of photosynthesis: temperature, light intensity, and carbon dioxide concentration. So light intensity, not just light, don't say light, and temperature. So now light intensity from here, from B to C, light intensity is not the limiting factor, but it could be temperature, it could be CO2 concentration, what could be the limiting factor. Then coming to question number 18, in which test tube will the breakdown of fat be the fastest? Now you can see there is the enzyme lipase is a question on the enzyme lipase. So there's lipase here, there's lipase here, there's no lipase here. And then there's boiled lipase. So boiled lipase means the lipase is now denatured. So this is not going to work. So there was no lipase here. This is not going to work. Now we are left with between A and B. Now it says, in which test tube will the breakdown of fat be the fastest? Fastest means it's going to take a little very short time. So you have to realize it has to be in this one. Why? Because bile emulsifies fat. So the large globules of fat, what does bile do? Bile converts them into smaller globules. Now, if they are smaller globules, then this is going to be the lipase will have more surface area to work upon. So the more surface area, so it will mean it will break it down faster. So that is why the answer is A. Milk, lipase, and bile, because bile emulsifies fats. Now, whenever you're doing this video, please remember to pause here, read the question yourself, and then think of the answer. And then, so you need to pause this video many times to be able to really fruitfully use this video as a learning tool. Question number 19, the diagram shows stages in the passage of water through a plant. Which arrow shows water moving in the form of water vapor? Which arrow shows water moving in the form of water vapor? Now, let's look at it. A, B, C, or D? C is, of course, the movement of water. So this is liquid water. So this is wrong. Then water moves them from the soil into the root hair cell. This is wrong too. So you have to understand why it is wrong. I don't want you to just learn it what is right, just learn the answer, or just memorize the answer or ratify the answer. No, you can't be doing that. So the question is that, you see, in B, what is happening? Water is moving by osmosis from one cell to the other. Here it is moving in the xylem in the form of a liquid water. Here also in the form of liquid water. This is the only place where it's not moving in the form of a liquid water. Why? Because it has evaporated and now in the shape of a water vapor, it is passing out of the stomata as a gas. So which arrow in the form of water vapor, liquid water or water vapor? So this is all, this is liquid water, this was liquid water, and this was liquid water. Yeah, this was in the form of vapor, so that is why the answer is B. The diagram shows some potato tubers, new shoots are beginning to grow, sucrose is being translocated from source to sink. So the tuber potato is growing and it's developing these roots and these shoots. Now, which statement is correct? Now, if you know what is source and sink, source means the provider and sink means the user. Source means the provider and sink means the user. So the tuber is a sink, the soil is a sink, the shoots are sources, the shoots are sinks. Now, the tuber is a sink. No, the tuber is actually the source. Why? Because it is giving out the starch stored as glucose and then the glucose being respired and these things are growing out of it. The soil is a sink. No, that's always a part of a plant which is a sink. So this is wrong. This is wrong. The shoots are sources. No, the shoots are actually growing out of this tuber. So the answer is D, that the shoots are sinks and the tuber is the source. So it's basically a provider. The tuber is a source, so it is the provider. Tuber sources are providers and sinks are the users. Now, question number 21, which statement about passive immunity is correct? Active immunity was when a person falls sick and then his lymphocytes make antibodies. So that was active immunity. And this is natural and artificial active immunity. Passive immunity is when ready-made antibodies are either passed from the mother to the fetus across the placenta or in the form of we prepare them in the lab and then we give them antibodies, like when you fall on the road, you have an injury to yourself on the road, or you graze your knee and you get a big cut there, and so some of the mud might have entered your knee. Then you get a tetanus injection because there are tetanus bacteria in the soil, and they might have entered your body, and we can't afford you to have tetanus. It's a very lethal disease. So the question is, it says antibodies are acquired. It gives a long-term effect. No, it's a short-term effect. The effect of the mother to fetus or the other artificial passive immunity also is short-term. It is inherited. No, memory cells are produced. No. So the only answers are antibodies are acquired. They are obtained from a source, either mother to fetus or we make them in the lab. A person eats a large bowl of rice. Rice contains starch. What happens to the amount of insulin and glucagon in their body? So what has to happen if you eat starch? Well, it's converted to glucose. Glucose enters your blood, so your blood glucose level rises. And where is detected? Detected in the pancreas, and that causes the release of insulin. So insulin is going to increase. And what will happen to glucagon? Naturally, glucagon is going to decrease. Why? Glucagon is only released when there is low blood glucose levels. Like for instance, when we are fasting, just before iftari time, that is the time when a lot of glucagon is being going to be released. That's why people use the word hungry instead of saying hungry, they say hangry, means that you are hungry and you are angry. So I always see there's a research needs to be done whether glucagon causes the anger or the hunger causes the anger. Then we come on to question number 23. What happens to the mass of DNA in nucleus before mitosis occurs? So you have 46 chromosomes in your one cell. Now the 46 chromosomes have to become 92 before the cell can divide by mitosis, because each cell that is formed has to have 46 chromosomes. So this is the point in which DNA replication has taken place. What happens to the mass of DNA in nucleus before mitosis occurs? So what we have to understand is that the answer is A, it doubles. The mass of 46 chromosome is say 2 nanograms, so it becomes 4 nanograms. So it doubles, it doesn't half, it stays the same, it halves and then halves again. No, that's for more for meiosis. Then coming to question number 24, which statement about variation is correct? Now, which statement about variation is correct? Continuous variation results in few phenotypes with no intermediates. What was continuous variation? Height in 16-year-old boys. So that is a many intermediates. Discontinuous variation is like four blood groups, results in few phenotypes with no intermediates. Yes, that is the answer B. But why is C not the answer? Phenotypic variation is caused by environmental factors only. Well, no. 16-year-old boys, why would they have different heights? Because number one, they had different genetics, and then with the different genetics, they got different environmental conditions. Maybe some got more protein to eat in their when they were in their teens, or somebody got less proteins, or somebody got more carbohydrates, more fats, more they got more better food, better health-wise food, not more food. I didn't say more food, I said better food. So phenotypic variation is caused by environmental factors? No. Phenotypic variation is caused by genetic factors? No. Actually, phenotypic features are caused by both environmental and genetic factors. Question number 25. Some plants of different species can be crossed with each other to form hybrids that have a diploid number different from either of the two parent species. The diagram shows a cross between plants with different diploid numbers. So species 1 diploid number is 70. And species 2, they've left this out. This is something which you've got to find out. Hybrid zygote diploid number is 64. Now you have to realize is that if this was going to divide by meiosis, then it would have 35 chromosomes, but half the number of chromosomes. And then in the zygote, then the total number here is 64. So what has come from the rest of it? So 64 minus 35. 29. So 29 have come from here, but the diploid number will be 29 plus 29. So what will that be? 58. Why? Because you see, what you have to understand is that from 58, so from 35 has come from here, and the half of 58 is 29. So 29 plus 35. 29 plus 35 is how much? 64. So that is how we've got this 64. So that is the answer for D. Species number diploid number X is D is equal to 58. Then coming to the question number 26. In the next question, the diagram shows some of the stages involved in the nitrogen cycle: atmospheric nitrogen, nitrate, plant protein, and ammonium compounds. Now, what we have to understand is that let's look at the first one. What is one? Nitrates to atmospheric nitrogen. So this has to be denitrification. This has to be denitrification because you see, nitrates to atmospheric nitrogen is denitrifying bacteria which live in waterlogged soil. Now, when that is, then it's very easy. There's only one of this, and everything else fits in. Okay, then everything else fits in. It's not very difficult now. So one was denitrification, two is absorption, right? Three is decomposition, four is nitrification, and five is atmospheric nitrogen to nitrates. This is nitrogen fixation, which is lightning. Nitrogen fixation, or which is the other word is lightning for it. How is nitrogen of the air fixed and it becomes nitrates in the soil? So this was how you had to be doing this question. Next question, what are the smaller basic units of starch and glycogen? We know starch is glucose and glycogen is glucose. So the answer is D. So it couldn't have been anything else. You see, glycogen is where you're going to get it mixed up is with fatty acids and glycerol. Some of you are going to get mixed up with that. Now that's not glycogen, that's glycerol. And that is why they kept all these, fatty acids and glycerol so as to confuse you all. Then coming on to question number 28. Now this is a little difficult. Okay, question number 28. A chromosome was analyzed and found to have 6125 pairs of bases. Pairs of bases means what? A, T, G, C, A, T. These are three base pairs. So 6125 pairs of bases. So If I said this is 3 base pairs, it means 6 nucleotides, 6 bases. So 6125 into 2, 5 to the 10, 0 carry 1 to 5, 2 ones are 2, 6 to the 12. So 12,250 total bases it becomes. Got it? 6125 pairs of bases. Just like I said, 3 pairs of bases means 6 nucleotides. So I multiplied by 2. 3 into 2 is 6. So I get a total number of bases is 12250. Now what do I do? What do I do? I have been told that the type of each base was identified and 2, 3, 4, 5 were G. So if 2, 3, 4, 5 were G, then 2, 3, 4, 5 were C, GC, ATGC. So this comes to a total of 4,690. So I have minus that from the 12,12250. I have minus this one from that, and this of course comes to be 7560. Now that means if I say G was 2345, then the C was also 2345. The remaining that is left is 4,690. Half of these are A and half of these are T. So 7560 was the remaining. Then 7560, I have to divide this by 2. And this is equal to 3780. Why? Because 3780 are A and 3780 are T. So that is why the answer is B. Please try to understand it. We said base pairs. So 6125 into 2 is this is the number of base pairs, right? Then 125 then you took then we were told that there was the G's. So there was 2, 3, 4, 5 of the G's. So add this is 2, 3, 4, 5 G, 2, 3, 4, 5 C. So add it 4,690 minus the 12250. This is coming from here. Now when you minus it, the answer is 7560. Now you have to divide this by 2. And the answer is 3780, and that is why the answer is 3780. Question number 29. The statements describe some events that occur in a plant after light energy is absorbed by the chlorophyll. Converted to chemical energy, converted to sucrose for translocation, starch is stored in the roots or seeds, and glucose is produced. Now, the first thing we've got to say, light energy is converted to chemical energy. So 1, then glucose is produced 4, then converted to sucrose for translocation, and then starch is stored in the roots or seeds. So that is why the answer is D. For question number 29, the answer is D. Very simple. Converted to chemical energy, glucose is produced, converted to sucrose for translocation, and then after quite some time, all that starch will be stored in the roots and the seeds. Now it says, question number 30, what is an example of passive immunity? Immunity against influenza after an individual has been infected with the influenza virus. No, you do not have any disease. Your lymphocytes, in fact, do not make antibodies. We get ready-made antibodies. So this one is wrong. The formation of memory cells, no, that is again wrong. Yes, this seems to be correct. Given chickenpox antibodies to people at risk of developing a severe infection, vaccination against measles were a harmless version of the virus. This is active immunity because your lymphocytes are going to make, so we give you the weakened virus. So this would be artificial active immunity. Now, 31, a young maize seedling is grown from a seed. The seedling is placed on its side and left in a room with a light source for several days. So the light source is coming from the top and the root is this side and the shoot is towards the right. What will happen to the young shoot? Yeah, yes, the shoot is going to grow upwards. So this shoot is going to grow upwards. Auxin becomes concentrated in the lower part, yes. And it causes it to grow more. So it is in the lower part. So auxin becomes concentrated in the lower part of the shoot and the shoot grows towards light. So what we have to understand, that is the correct answer. But why are the others wrong? Let's talk about that. Auxin becomes concentrated in the upper part of the shoot. No, that's again wrong. It's away from light. Auxin is evenly distributed in the shoot and the root grows toward. No, not that's not in the case. Auxin is evenly distributed in the shoot and the shoot grows away from the light. It doesn't go away from the light. It's going to grow towards light in this situation. Okay, coming to the next question. What is an adaptive feature of sperm? Large stores of energy, enzymes in the acrosome, jelly coating that changes the fertilization, and a diploid nucleus. Well, that is wrong. Diploid nucleus is never there. So what we have to understand is jelly coating that changes after fertilization. Well, no, the nucleus just enters at fertilization. Enzymes in the chromosome, yes, that's correct. And large stores of energy, no, in fact, the ovum has large stores of energy, not the sperm. The sperm just has to get there, nucleus enters, and then the nuclei fuse and forms the zygote. 33. The graph shows the levels of two hormones during a 28-day menstrual cycle, concentration of hormone, time of the days. Which hormones are represented by X and Y on the graph? Naturally, it's estrogen and progesterone because LH would peak here. There'd be a graph like this. And FSH would be, of course, higher here and then it'll go down. So, what we have to understand is that which hormones are represented by X and Y. So, this X is estrogen and Y is the progesterone. Question 34. When a horse with red hair, HR breeds with a horse with white hair, HW. The offsprings have red and white hair. Now you can see this is a phenomenon of codominance, red and white hair. These horses are described as having roan colored hair, roan, R-O-A-N. This is an example of codominance. When two horses are roan, roan means what? HRHW. So when two horses are roan and they're bred together, what is the chance of the offspring having roan colored? So this is the offspring. So 50% out of the 100%. This is 25% and this is 25%. And the roan is 50%. So that is why the answer to question number 34 is C. Which statement about selective breeding is correct? Question number 35. Which statement about selective breeding is correct? It does not involve humans. It involves a struggle for survival. It always involves only one parent. It involves parents that possess the desirable features. So that is the main thing about selective breeding. It does involve humans, it involves a struggle for survival, it always involves one plant. No, we actually choose the male and the female and then we cross them and then we do this over many generations and that is called selective breeding. So the answer is D, it involves parents that possess the desirable features. Question number 36. The diagram shows part of the carbon cycle. So we have carbon dioxide in the atmosphere, we have this going out of it. So this is going to the producer, then we have fossil fuel and it's going back. So that's combustion. Then we have decomposers, and then we have consumers, and the consumers are returning carbon dioxide, so that means respiration. And so which process shown by the arrows labeled X? That's D, respiration. All of these organisms, the bacteria and fungi who are the decomposers, they respire. And then the consumers, which is the animals, birds, they will respire. And the producers, the plants also respire and return the carbon dioxide to the atmosphere. So that is why 36 is D, respiration. Then coming to 37, which process makes use of a genetically engineered organism? Using bacteria to produce insulin, using enzymes in biological washing powders, using bactinase in fruit juice production, using yeast to produce ethanol. What process makes use of a genetically engineered microorganism? So 37. The answer is A. Why? Using bacteria to produce insulin. That is genetically engineered organisms. Using enzymes in biological powders is not genetic engineering, or pectinase in fruit juice is not genetic, as biotechnology. Using yeast to produce ethanol, no, this is not genetically engineered organisms. Question number 39. The cholera bacterium produces toxins that cause chloride ions to be secreted into the small intestine. What is the immediate effect this on the water potential of blood in the intestinal capillaries and the water potential of the contents of the small intestine? So if the chloride ions are going to be moved from the blood into the intestine, so more chloride ions into the small intestine, so the water potential would be lowered. And what will happen to the blood? That will be raised. Why will it be raised? Why would it be raised here in the blood? Because the chloride ions are coming out of it, so there are more free water molecules. So more free water molecules means that the water potential would be raised. You see, more the free water molecules, more the water potential. More chloride ions means less free water molecules. So when the chloride ions are in the intestine, then of course the intestine's water potential will be lowered. But when they're coming out of the blood, then there are more free water molecules now in the blood. So that will be raised. So that is why the answer is C. Then coming to question number 40, the graph shows the concentration of maltose in different parts of the alimentary canal. So in the mouth, in the stomach, and in the small intestine, you can see how the maltose is increasing. We see it increasing here, then we see it leveling out, then increasing, and then we see it decreasing. What causes the change in concentration at X? What is happening to the maltose level? The maltose concentration is decreasing. Question number 40, when you can see when X, what is happening? The maltose is decreasing. Now, maltose is a disaccharide. It's made of two glucose units joined together. It's two glucose units, and maltase, what does it do? It's going to break it up, and then the glucose is going to be absorbed into the blood. So, absorption of maltose was wrong. Action of amylase was wrong. What was correct is action of maltase. So, the maltose concentration, whatever the maltose was present, is now being broken down into two units of one glucose unit and another unit of glucose. So the level of glucose would increase in the small intestine. It would further increase. You see, in the mouth, yes, maltose starch will be converted to maltose. Then in the stomach, nothing's happened to maltose. Then the more and more in the small intestine, there is more amylase working on the starch to convert it to maltose. And then we have the final enzyme called maltase, which is going to break down the maltose into two glucose units, and each maltose will give you two glucose, and then that glucose will be absorbed into the blood. Question 38, the two test tubes are filled with hydrogen carbonate indicator. An aquatic plant was placed into each test tube, and the test tubes were sealed with bungs as shown. Lamp, hydrogen carbonate indicator, so that means photosynthesis will take place, so CO2 is going to decrease. Here this is enclosed in excludes light, so respiration is going to take place, and there is going to be more CO2. Test tube X was illuminated, test tube Y was kept in the dark. The results are shown. Color of the hydrogen carbonate indicator X was orange at the start, and then it became red and orange and yellow. What causes the color change in the hydrogen carbonate indicator in X and Y? So the answer is A, a decrease in the concentration of carbon dioxide and increase in the concentration of carbon dioxide. And these colors are something all to do with carbon dioxide. The carbon dioxide color more or less is going to determine the final color. This is the first video on these mock exam test questions, and another video follows this which will contain more questions on the difficult questions on these two syllabuses.